Thanks so much. And I'd like to also thank Jenny for her talk. Our talk is going to cover many of the same questions, and just looking at a different area in the brain. As Sven said, we're going to be looking at the midbrain, yeah, we're also going to see Emily Allen and Andrew Oxenham again in this talk. So today I'd like to talk about, as I said, neural mechanisms for timbre, and specifically spectral-centroid coding, and we're specifically looking at the midbrain. And I'm going to start by introducing those two topics and talk about our computational modeling approach. First, I just want to really quickly review what the spectral-centroid is. We've heard a lot about it at this conference so far. But it's generally a measure of where the bulk of the energy is in the spectrum. It's calculated as a weighted average frequency where the magnitudes, the magnitudes of the frequency components are the weights. Here you can see in this example from Stephen McAdams, two spectra, an oboe and a trombone. And you can see that all other things being equal, the oboe would have more energy at higher harmonics and the trombone at lower harmonics. And so we say the oboe has a higher spectral centroid, the trombone has a lower spectral centroid. And the descriptor of spectral centroid has been associated, again, as many people have said already in this conference, with the perception of brightness. So in this talk, we're looking at the midbrain, which is earlier. It's also called the inferior colliculus. And it's a really interesting place to look at because it's early in the auditory system, but it's also obligatory, which means that all of the information that's coming from the ear to the cortex has to pass through here. So all the information is here, is in the midbrain somehow. It's just a question of how. Okay, so we have the midbrain. Here's our computational modeling approach. We have 4 steps. First, we replicated stimuli, and these are actually from one of Emily Allen and Andrew Oxenham's experiments in 2014. This particular study tested how far apart spectral centroids of 2 stimuli need to be for listeners to correctly identify which spectral centroid is higher. And I'll describe that more in a second. Next, we plugged these stimuli into computational models that simulated neural representations of the spectral centroid, first in the auditory nerve and then in the midbrain. And finally, we used these model responses to simulate listener performance in Allen and Oxenham's experimental paradigm. So I'll start by talking about the stimuli. These were complex tones with a single peak in the spectral envelope. Here I've shown it on a linear frequency axis so that you can see the fundamental frequency is present as the difference between each pair of harmonics. Here I've shown the same stimulus on a log frequency axis, and so that you can more clearly see the single spectral peak in this stimulus. And I'm going to refer to this following Allen and Oxenham as the spectral centroid of the stimulus, although obviously the exact value of that would vary depending on the method used to calculate the centroid. All right, so on each trial of Allen and Oxenham's experiment, the listeners heard 2 stimuli, one with a lower spectral centroid and one with a higher spectral centroid, and their task was simply to identify the one with the higher spectral centroid. The experimenters gradually estimated the smallest amount by which the 2 could differ, such that the listeners would get the task correct 71% of the time. Interestingly, Allen and Oxenham also found that when they varied the fundamental frequency on each trial, as well as varying the spectral centroid, it became more difficult for listeners to do this task. And the more, the x-axis here shows increasing variation in fundamental frequency. The more the fundamental frequency was varied, the harder the task became. So this gets exactly at the question that Jennifer Bisley was just talking about, the question of kind of whether percepts are invariant or whether they are represented, I should say, whether they're represented in the brain in kind of invariant ways, or whether the representations might reflect both timbre and pitch, for example, in the same neuron. So one explanation for these results is that the listeners just became confused as the variation in a different parameter increased. It was just simply a more distracting case. But we wanted to look and see if perhaps there was a basis for this interference in the midbrain representations themselves. So this is very similar to those neurons that Jenny was talking about, where they represent both timbre and pitch in the auditory cortex. We're kind of looking to see whether that might happen in the midbrain. All right, now moving on to our simulations. We used models that, based on a large number of known properties of the auditory system, simulate how these stages of the system might respond to a given sound. That sound can be arbitrary. So the first part of our model setup simulates neural activity in the auditory nerve, which is the bundle of neuron fibers that carry signals from the ear to the brain. Or sorry, I should say from the ear to the cochlear nucleus. The auditory nerve model we used is very similar to other models of the peripheral auditory system in that it uses a bunch of bandpass filters to simulate the function of the cochlea. But our model also, and when I say our model, it's a model from our lab, and specifically I'm using this, the version developed by Chelani in 2014. And this model simulates a number of physiologically realistic aspects of the process that are a bit more complicated. For example, the filters here are asymmetrical. The higher ones are sensitive to wider ranges of frequencies in terms of hertz. But one especially relevant aspect of this model is that the filters realistically become much wider as the sound level increases. So here's an example of how this actually happens in the auditory nerve. You can see here, this is from Rose et al. And they're recording from a single auditory neuron. The x-axis indicates different frequencies of simple tones that they use to stimulate the auditory nerve, and the y-axis indicates the amount of auditory nerve response. And the different curves show experiments at different sound levels. And you can see at relatively low sound levels, 40 dB SPL, the auditory nerve fiber responds over a pretty narrow range of frequencies. But once you get up to the sound levels used, for example, in the Allen and Oxenham experiment, this 70 dB SPL, which are also closer to sound levels really used for music listening, these filters become much broader. So for example, this single auditory nerve fiber is responding over almost a span of a kilohertz. So here's a quick schematic description of what this looks like in our model. At every moment, the model evaluates the incoming sound, and at lower levels, the filters are very narrow. And then as the level increases, the filters become broader. All right, so this is one type of result that we get from the auditory nerve model. These are average rates across a tonotopic population. So the x-axis here indicates the cochlear region from which the auditory nerve receives input. The y-axis indicates the activity of the neurons. So higher on the axis is more activity. And as you can see, at very low levels, represented by this black curve, there's a very clear representation of the centroid of this stimulus. However, as we go up to higher levels, like 70 dB SPL, the representation becomes very broad and really not a good representation at all. And this is one problem with rate coding, particularly in the auditory nerve. I'll mention here we ultimately were interested in extracting an estimate of the spectral centroid from these representations. To do that, we cut off things below this red line in order to cut off some of the spontaneous rate. And then we take a spectral centroid estimate just using a typical kind of weighted average procedure, just the way it would work with a regular spectrum. So here are centroid estimates. You can see centroid estimates very reliable at low SPLs, but then at higher SPLs, it wanders quite a bit. All right, so the auditory nerve model also produces timing information. And specifically, this model produces an estimate of how likely an auditory nerve fiber is to fire at every moment in time during and after the stimulus. So watch out, the x-axis here has changed to time. And we can see an interesting pattern. So this is on the left, I've shown the stimulus that led to these responses. And you can see at the spectral-centroid, there are there's no slow temporal fluctuations in the envelope of the auditory nerve response. But anywhere away from this spectral-centroid, there are a lot of slow fluctuations. And these are caused by beating between components, not necessarily a perceivable beating, but mechanical beating within these channels. The periodicity of this beating is equal to the difference between components. So in this case, with a 200 Hertz fundamental, the beating is at 200 Hertz. And again, these don't occur right at the centroid because this locally prominent peak dominates and eliminates the beating. Okay, so now we're going to take this temporal information and pass it into the midbrain model. And here we focused on on the responses of one type of midbrain neuron called a band suppressed neuron. These neurons increase in activity when they receive input that does not have slow temporal fluctuations. And they decrease in activity when they receive input that does have these slow fluctuations. And it's kind of interesting. This is one of the first places in the auditory system where this sensitivity to these kind of fluctuations happens. I will mention one thing just to connect to Jenny's talk. This pattern, since it's sensitive to these slow fluctuations, this rate pattern in the midbrain takes around 40 milliseconds to become sharp. So I know we were talking about earlier in the last talk how to get 70 milliseconds worth of useful timbre information. And that might be, I don't know, that's an interesting thing to think about. Okay, I'll flip this graph here so we can see it more easily. And here we can compare the stimulus and the rates in the auditory nerve and the rates in the midbrain. And you can see that the midbrain is not only a sharper code, but it's also stable across different sound levels. Again, because it's taking advantage of that timing information in the auditory nerve. It's also, I'll also mention here, although we've represented this, and we talked about this in terms of the midbrain rates. This is just one example of how one neural system might kind of read out timing information. And since the basis of this sharp code is the timing information, it's quite possible that the midbrain is not actually the ultimate readout location of this information, or maybe there really is no readout. That's important to mention as well. So you can think about this type of sharp spectral-centroid code as really a property of the temporal information. All right, so now we're familiar with this midbrain representation of the centroid. The next question is whether there's a basis for changes in fundamental frequency to kind of interfere with this representation. And we're interested in this because we see that interference effect in the Allen and Oxenham data. So we're interested in just seeing if there's anything that could explain that. And because this code is not just a sharpened representation of the centroid, but actually a sharpened representation of harmonics near the centroid, this code does shift as the fundamental frequency changes. So you can see there's this movement in this primary peak from left to right as we increase the fundamental frequency. However, the actual centroid estimation is more complicated because representation of other harmonics changes over time. We're looking at a couple different ways of potentially estimating the centroid from this information. This is kind of a work in progress, but it is clear that the fundamental frequency can impact this timbre code. Let's move on to simulating the listener responses. So basically all we did to
Braden, if I may interject, I'm just letting you know that you're already past 15 minutes. So if you could keep that in mind, thank you.
Thank you. So yeah, basically we just put the model in the same situation as the listeners, ask it to give us a centroid estimate. And when we do that without any variation in fundamental frequency, the model actually does a really good job of arriving at the same threshold as the human listeners in their study. When we add in fundamental frequency variation in the same way that they did, we actually, despite the complexities in the ways that fundamental frequency interacts with timbre in this midbrain code, we actually see a really similar increase in threshold based on the model results. And there's no kind of free parameter. We're not fitting this line to the human results at all. So it's interesting that it has the same trend and is in roughly the same percent correct, or sorry, has roughly the same difference alignments as the humans. Yeah, and I can talk about more future directions in the questions if there's time. Thank you so much.
Thank you, Braden, for a very interesting talk. I haven't seen any questions in the chat, but I do see that Asteris has a question. Oh no, that's a clap, sorry. I misread the symbol. But there, I see some activity on the chat now. So Kai has a question for you.
Peter, you want to go first? I think it's your turn.
Sure. We did a study of vowel category boundaries directed by psychoacoustician Tatsuya Hirahara and with Bertrand Delgut in the 90s. And if you look at the population interval distribution of the population-wide interval distribution of interspike intervals, all order ones in the auditory nerve, you can predict the vowel boundaries very closely. And they're very, very sharp. Within 10 hertz, the vowel category changes. It wasn't clear to me what the relationship of the timing in the auditory nerve to these band-suppressed neurons is. You reference it, but when you're talking about temporal information, are you talking about fine timing information or envelope information or what?
Yeah, that's a great question. Thanks. We're talking about envelope information, envelope of the response, and I always try to distinguish that from envelope of the stimulus itself, because once the envelope is the stimulus is bandpass filtered, then everything kind of changes. But yeah, we're talking about the envelope of the response and slow timing information. And
on the order of what kinds of periodicities in terms of hertz?
In this case, 200 hertz.
Okay.
Mm-hmm.
So I think one quick question from Kai then maybe, because
yeah, just one comprehension question.
To move on.
If you go to the next slide, Braden. So it's really interesting to see how the midbrain sort of develops this potential selectivity, or how one could call it. But I was just curious, for 50 dB SPL, how do you, or what do these additional spikes mean?
Yeah, that's a great question. So these additional peaks are other harmonics, the representations of other harmonics, and we see that pretty frequently. I mean, one of the things we didn't get a chance to talk about in this presentation was that in Alan Oxenham's experiment, these stimuli are moving around quite a bit, and there's a lot of changes going on. And so we do see this pretty often. other harmonics. And the spectral-centroid encoding mechanism works, the same way. It's just that when there's multiple prominent peaks, those multiple peaks play a role.
Okay, thanks.
Okay, thanks again, Braden, for a very
Thank you so
interesting talk. And we'll have to move on to the next talk, which will be delivered